Hi everybody, it's Ajanette and I'm here to go over your Chapter 2 lecture on neuroscience. So neuroscience is the intersection of biology and psychology. There's three main branches that we consider in psychology. Behavioral genetics studies the relative contribution of genes and environment, and they end up producing heritability rates and concordance rates. Heritability rates are estimates ranging from 0 to 1 or 100, and they represent the estimated contribution of genes to any particular behavior or traits. The second field of the neuroscience is evolutionary psychology, and the second branch of psychology that focuses on the relationship between biology and psychology is evolutionary psychology. This particular field tries to explain behaviors or traits or trends based on what the adaptive value of that particular behavior or quality is and how it may have contributed to the survival of the species. And then neuroscience, which is kind of the main focus of this chapter, is also called biopsychology or psychobiology. You hear it kind of used interchangeably. Most often neuroscience is in a psychology department, but it can also sometimes fall into a biology department. But it very much is interdisciplinary in that people from multiple specialties or professions come together and they study the underlying biological processes involved in disorders and mental behavior, psychological functioning, and some of the behavioral issues that are studied in psychology. One key area of neuroscience is that of genetics, where we look at chromosomes. So as human beings, we have 46 chromosomes, 23 pairs. Each of the pairs represents a chromosome from mom and a chromosome from dad. And those chromosomes are made up of the building blocks, which are our genes themselves. And the combination of the genes essentially represents our template, or the code that our body lays out for our genes, kind of like the blueprint for how we're going to develop, not just physically, but possibly even psychologically and even potentially in terms of our behavior. It was once thought that all gene pairings, because every gene pairing one comes from mom and one comes from dad, and these genes can either be dominant or recessive, where if a dominant gene is present, it automatically overrides the recessive gene. It was originally thought that all expressions of genes work in this dominant inheritance pattern such that the one dominant gene involved in that pairing would automatically make you express the dominant version of that gene. So take, for instance, me, I have brown eyes. Brown eyes is a dominant gene. However, my father has blue eyes, so blue eyes is a recessive gene. When you have a recessive trait, the underlying genetic coding has to be too recessive. You had to have received a recessive from mom and dad in order to have that expression of the recessive gene. So blue eyes is recessive. My dad has blue eyes. And so all my dad has are two blue-eyed genes to pass on to me. And then I know I have a dominant gene from my mom because I have brown eyes. So what we call our underlying genotype can have any combination of dominant or recessive. For a dominant expression, it can have two dominant genes, it can have a dominant or recessive gene, and then for recessive traits, you can only have two recessive genes. Although we thought that everything worked on a dominant recessive inheritance pattern, we actually now know after the Human Genome Project that things are often polygenic, meaning multiple genes contribute to the expression of any particular trait or characteristic. So if we found a particular disorder that was a recessive disorder, theoretically, we can go in and manipulate those genes and take out the recessive gene. But because things are polygenic, taking out the recessive gene for one particular trait or quality may actually produce problems in other traits or qualities, because it might have a role in that particular trait, but roles in other traits as well. So the bottom line is that although our knowledge of genes has expanded significantly, we realize that we still are not able to progress forward in the manner that we thought we were going to be able to progress forward understanding the Human Genome Project. So, okay, so let's look at some ways in which behavioral genetics studies the relative contribution of genes and the environment. First of all, they engage in twin studies, and they compare the rate of shared traits or tendencies between monozygotic twins, which have 100% of the same DNA and therefore are 100% genetically identical. And then they compare them to dizygotic, which means they came from two separate eggs, and they share just as much DNA and genetic similarity as any other siblings. And we can see that if traits are more similar for monozygotic twins, then genes are likely to play a higher role in that trait because they are 100% genetically similar. Whereas if being a monozygotic twin doesn't in any way increase the likelihood of you sharing traits or qualities, then genes aren't necessarily a factor. We also look at family studies where we might look at concordance rates, where concordance rates look at what is the shared similarity between you and a family member, and as you increase in genetic similarity to those family members, do the genetic similarity rates increase as well? In other words, are you more likely to be genetically, or are you more likely to be similar to a sibling on behavioral traits or qualities, or a cousin, let's say, relative to the general population? And if we see that rates of shared experiences are increasing, our rates of shared tendencies are increasing based on genetic similarity, then we're more likely to assume that genetics plays a much larger role. Then additionally, we can look at adopted children, and we can study adopted children in terms of, are they more similar to their biological parents, or are they more similar to their adoptive parents? They're more similar to their biological, we assume genetics are a larger role, whereas if they're more similar to their adoptive, we assume the environment plays a larger role. And then finally, disorders and diseases, where we look at what is the concordance rate, or what is your risk or probability of obtaining a particular disease based on the occurrence of that disease in a family member, and does that increase again with genetic similarity of that family member? Finally, behavioral genetics produce what we call heritability rates, which is again the estimated percentage of the contribution of genes to any particular trait or circumstance. They range from 0 to 100%, 100% being that it's fully genetic. So let's move on to neurons. Neurons are the basic cellular structure of the nervous system. We have over 1 trillion neurons in our body. They're not nerves. Often we think of neurons and nerves simultaneously, but they aren't. Nerves are actually bundles of axons, which are parts of neurons, and they can run the entire span of your body. Each neuron has kind of an individual function and role, and those vary from neuron to neuron to neuron. So not all neurons work the same. There's also glial cells that make up a large part of our nervous system. And right now we view them more as just support cells, where they help to maintain and protect the neurons or insulate them. And then they also help to remove the waste and provide nutrients and support to the neurons. Interestingly enough, more recently, research has shown that they actually do play a more active communication role within the nervous system. They can actually activate action potentials, which we'll talk about in a little bit. So they may actually be much more active, where we looked at them more as background singers in the past. They might be much more active than what we realized. So we still have a lot more to learn about glial cells. So here we have a diagram of the structure of a neuron. And so here's your soma, or your cell body. Within the soma, you have the nucleus. And down here is the axon hillock, where the soma starts to kind of merge into the axon. And then this is the initial segment of the axon. You'll see the axon spans the entire length here. At the end of the axon, it branches out into branches, and they're called terminal buttons here. Inside, there's vesicles, which are sacs that contain neurotransmitters, and the neurotransmitters are the chemical messengers that are released from the neurons. And then that's going to neighbor and partner up with another neuron who's going to be close by through the extension of what we call a dendrite. The dendrite are the receiving branches of the neuron. So the dendrite picks up the chemical messenger, processes it, tells the neuron what to do with that chemical message. It goes here, and it's what we call summated, where it determines whether or not we have enough information to fire or not fire, passes that message, and then it's going to go down the axon, assuming it's told to fire, come out here to the terminal button, stimulate the release of neurotransmitters in that synaptic gap, or that space between two neurons, and then the process starts all over again in the next neuron. So neurons are, they communicate two ways within the neuron. They communicate via action potential. It's an electrical charge that's sent down the axon of the neuron, assuming that it's received enough excitatory messages. And then we also know that, excuse me, I went the wrong way. Then at the end of the axon, it's going to release neurotransmitters from the terminal buttons over here. And neurotransmitters are the chemical messages. And keep in mind, those are spread from neuron to neuron through that synaptic space. So we communicate chemically between neurons and electrically within our neurons. So that primary electrical communication is facilitated through what we call action potential. And that's essentially sending an electrical nerve impulse down the axon of our neuron. And then so those are kind of the two primary ways our nervous system communicates within, again, through action potential, between, again, through chemical exchanges. And so our neurons are really communicating from neuron to neuron, but our endocrine system is a much more global communication system. This is where hormones are pumped into our bloodstream. Since our blood circulates throughout the whole body, everybody knows how much of any given hormone I have in my blood system at any given time. So it's not from cell to cell, it's kind of to the masses, kind of like a Twitter post as opposed to maybe an individual email. Action potential is an interesting concept. If you've taken a chemistry class, you might have a really good understanding of it already. But we look at all ions, in this case specifically sodium and potassium, and they have either a positive or a negative charge, and calcium and sodium both have a positive charge. And so what happens is, you probably heard opposites attract. They can also to some extent repel one another, but either way, your neuron is trying to maintain a balance of positive and negative charges. When it's at rest, it's not really doing anything. It's called resting potential, and it's potential because it always has the potential to generate energy, but as of right now, it's not. It's just resting. And so that's when you see that the ions are polarized. They're kind of on one end or the other. They're in or out of the neuron. And you have a negative charge inside the neuron and a positive charge on the outer membrane of the neuron. And along the neuron, there's membrane channels that are going to open or close and let ions pass through. When action potential is initiated, so when the soma of the neuron gets enough excitatory impulses to say, okay, I'm going to fire, then those gates start to open up like a wave along the axon. And so one opens, one closes, opens, closes, opens, closes, opens, closes, opens, closes, all the way down the entire length of the axon. And